All right, welcome. Today, we are diving into the absolutely revolutionary world of molecular docking. We're going to be on a quest to find the perfect key for a very, very specific microscopic lock, a search that could honestly unlock the next generation of life-saving medicines. So, to really get why this is such a game changer, you first have to appreciate the problem it's trying to solve. Finding a new drug is, well, it's unbelievably hard. It's so time consuming, and it costs a staggering amount of money. We're talking billions of dollars, and sometimes more than a decade of work, just for one successful medicine. Here's a great way to think about it. Imagine a disease is caused by some protein in your body that's gone haywire. That protein is the lock. Now, to fix it, you need to find a tiny molecule, that's our key, that fits into that lock perfectly and just stops it. The problem? You're looking for that one single key in a pile of billions of possibilities. And billions is a huge number. If you tested one new key every second, all day, every day, it would still take you over 31 years to get through them all. No joke. And yeah, that's the heart of it, really. It's what we call the $1 billion search. The old school way was to just manually test thousands or even millions of compounds in a lab. It's a total brute force approach that's just super slow and inefficient. But then, someone asked a question that changed everything. What if, instead of just randomly trying keys till we get lucky, we could get a detailed 3D map of the lock, the protein, and use a computer to actually design a key that we already know should fit? And that right there is where molecular docking comes in. It's that digital shortcut we were just talking about. It's this powerful computational method that lets scientists model that whole lock and key thing right inside a computer, which, as you can imagine, saves an incredible amount of time and money. Okay, so here's the crucial thing to remember. Docking is all about prediction. It takes the 3D structure of our protein lock and a potential drug key, and it uses some serious computing power to predict the most likely way they'll stick together. And just as important, it tries to predict how strong that connection is going to be. Now, you might think this is some brand new tech, but it's actually been around for a while. The very first algorithms were developed way back in the 1980s. But it was really the explosion in computing power in the 2000s that took this from a kind of niche academic idea and turned it into an absolutely essential tool that's used in, well, pretty much every modern drug discovery program today. So, how does it actually work? I mean, how does a computer find that perfect fit? Well, the process really has two main parts. First, the computer has to explore all the possible ways the key can physically fit into the lock. We call this the search. You can think of the search algorithm as the engine of this whole process. It's what does all the heavy lifting. It systematically twists and turns the drug molecule in every conceivable way inside the protein's little pocket. Its one goal is to find the best possible orientation, which scientists call a pose. Now, this search can happen in two main ways. On one hand, you have rigid docking. This is where the computer treats both the key and the lock like solid, unchanging puzzle pieces. It's fast, but, it's not super realistic. Then you have flexible docking, which is way more advanced. It actually lets the key and even parts of the lock bend and wiggle a little bit, just like they would in real life, which gives you a much, much more accurate prediction. Okay, so the search algorithm has given us a whole list of possible poses. But how do we know which one is the best? How good is that click when the key actually fits into the lock? Well, that's the job of the second major component, the scoring function. Think of a scoring function like a judge at a competition. It looks at every single potential pose the search algorithm found, and it gives each one a score. This score is based on a whole bunch of calculations that predict how strongly that key is going to bind to the lock. The poses with the best scores, those are your most promising drug candidates. And there are basically three main ways this scoring gets done. First up, you have the force field-based method. This is pure physics. It calculates the raw forces of attraction and repulsion between every single atom. Then in the middle, you've got the empirical approach. This is more like using a rule of thumb. It adds up points for things we know are good, like hydrogen bonds, which are like tiny little magnets that help pull the key into the perfect spot. And finally, there's the knowledge-based method. This one is really clever. It learns from a huge database of real-world molecules that we already know bind well, and it looks for similar patterns in our new poses. So you've got the search and you've got the score. That's the process in a nutshell. But how well does it actually work in the real world? Let's take a look at the docking scorecard, weighing both its huge successes and its remaining challenges. So, what are its superpowers? Well, there are a ton. Its main power is in what scientists call lead identification. That's just finding brand new keys that might work. It's also amazing for lead optimization, which is taking a key that's pretty good and figuring out how to tweak it to make it perfect. But beyond just finding drugs, it's also used to understand how proteins talk to each other and even how they interact with our DNA. But for all its power, docking still has its kryptonite. Getting that score, the binding energy, exactly right is still a major challenge. In properly accounting for how an entire protein can flex and move around, yeah, that's really tough. And believe it or not, even something as simple as the effect of water, which is literally everywhere in our cells, is incredibly hard to model accurately. But even with those challenges, the main takeaway is crystal clear, and this quote just nails it. Molecular docking isn't some neat little trick anymore. It has become an absolutely integral, you might even say indispensable part of how we discover new medicines today. And that leaves us with a pretty fascinating thought to end on. As these computational tools get more powerful and more accurate every single year, you have to ask yourself, could the next big life-saving cure be discovered not by mixing chemicals in a test tube, but by a brilliant algorithm finding the perfect key, all inside of a computer? It's pretty amazing to think about.